So we as a society are honored today to honor Tony Wright, and I'm very honored to be able to introduce him to you all. And Tony started his academic career on the West Coast. He was a California boy, and he kind of surfed around the waters there for a while, and he ate a lot of avocados and surfed and did a sports car thing, and he ended up at Stanford University. And one of the people that influenced him the most there was Gordon Bauer. And from there, one of the conversations he had was, you need to check out Columbia University, if I remember this correctly. And so also at Stanford, a very important event happened, and Tony met his wife Susan, who's here today also. And Tony and Susan, they didn't sail quietly around from Stanford to Columbia, but eventually they got on a sail ship at some point and lived there in New York City for a while. And so Tony was at Columbia. His initial sponsor there was Bill Cumming, who passed away when Tony was there. Tony Nevin supervised him from there. So after graduating from Columbia, Tony went to Austin, Texas, where he was a professor for 1 year, but Tony and Susan were like, they missed the big city, so they decided to go to Houston from there. And Tony's been there ever since. He was at the Health Science, the Sensory Science Center there for several years, and then he went to the University of Texas Medical School at Houston, where he's been there in the Department of Neurobiology and Anatomy since that time, 1995. Along the way, Tony's done a number of distinguished things. He was an NIMH pre-doctoral fellow at Columbia. He had a Fogarty Senior International Fellowship at the University of Auckland. He won the Holbrook Prize in 1989 to go to Germany and study there. And recently he became a fellow of the Society of the Experimental Psychologists. He's reviewed for way too many journals to count here, as you can guess. I think one of the most distinguished things about Tony's career is he's had about 40 years of straight funding from different sources, NIH, NSF, Army. And I think I'm just into the good stuff here. So one of the most distinguished things I think of Tony is the number of postdoctoral fellows and the visiting scientists that have come and studied with him over the years. And this is not a complete list, this is just a selective list. Peter Uccioli, Steve Sands, Skip Kendrick, Melissa Cheyenne, Julie Nyworth, Ramesh Bhatt, Masako Jitsumori, Jackie Emerton, Julia Phillips, Maria Alvarado, Jeff Katz, Ken Lysing, and Bob Cook. Tony's had over 100 publications. They range in a number of variety of journals that have impacted our field over the years. JAB, JEP, Animal Behavior Processes, General, Journal of Comparative Psych, Psych Bulletin Review, Psych Science, Current Biology, and Science. I think one of the things that's distinguished Tony's career is he often asks the question, he has a question, something he wants to study, and then when he figures out what that is, then he builds an apparatus. And as opposed to having an apparatus and then doing an experiment with that, he's always does it the other way around. And I think some of his most important work has been based on that logic. And it started at Columbia. He built his own operant chamber there to look at color functions in pigeons. It was affectionately named Taj Mahal. It's made out of mahogany, and if you ever see pictures of this, it's probably the sweetest operant chamber you will ever see. Of course, he's done a number of interesting things, and interference has been at the heart of his research all through his career. And the list memory work that Jocelyn introduced has been one of the hallmarks of that, and then recently developing the change detection procedures. So with that, I would like to welcome Tony Wright.
Thank you, Jeff, for that wonderful introduction, and thank all of you for your terrific presentations. In fact, so many things were covered better than I could have said them. I will only have to fill in the holes, so that's just terrific. So it's a great honor to be chosen to give this year's master lecture, and when I asked the people who nominated me what I should present, the answer I got is that it was requested that I organize my presentation around a theme that has guided my work, hence the title. And I include some of my earlier work, which some people may be unfamiliar. And finally mention people that have influenced you and your work. Jeff has already mentioned some of those individuals, many of them, as you'll see. So with these requests, I am going to begin mentioning some of those individuals, but I won't be able to go over that very quickly here. And then what I'll do is to briefly mention some work I did in psychophysics. And that's really sold me on what I'm going to call a functional relationship approach, which is systematic manipulation of critical variables. Some of this that you've seen in experiments that others have done or that we've done together. And then I'll show you how I've used this functional relationship approach to study and compare learning and memory processes. So, yes, I arrived at Stanford University. Gordon Bauer was my mentor. In fact, I took my first experimental psychology course in 1962 from Gordon Bauer. One of my good friends pointed out that, gee, this is, looks like a 50th anniversary here. And Gordon Bauer, who's very well known in human learning and memory and math modeling, had a rat lab, and we had a course with rats and experiments, and just to show you that he worked in animal learning and with rats. Here's Gordon Bauer. What would I need to do? Sorry, I need to set my, I can get it right here, just to set my arrow options visible, so I have a little pointer. So this is Gordon Bauer and his mentor Neil Miller at Yale University, and they're doing this self electrical stimulation studies that became very famous. And so he had just arrived at Stanford, gosh, a couple of years before I arrived there, so he was a young assistant professor. And I hooked up with him. I did a project in human memory with him. He was starting to do human memory work. And then he advised me, since I wasn't sure which area of experimental psychology to go into, to go to Columbia, as Jeff pointed out, Wright at Schermerhorn Hall. And Columbia had two areas, main areas of experimental psychology for the graduate students. One, visual psychophysics, headed up by Clarence Graham, very famous psychophysicist, and learning. In fact, Herb Terrace had just arrived either that same year or just the year before. But my sponsors, well, in animal learning, the people that I worked most closely with, as Jeff mentioned, were Bill Cumming and then Tony Nevin. So all the students, like in most departments, you'd work in one area or another, and with very little communication, but being eager and to learn all about these things, and the rest, I'm going to do it both. You know? So and this wasn't anything related to what my sponsors were doing or anything. So, you know, I studied psychophysics with Clarence Graham. He just published his book, Vision and Visual Perception. It's still a very well-known book today. And one of the things that impressed me, and this is just an example, it serves as a little prototype for the kind of things that I wanted to do, were some of the functional relationships you saw in psychophysics. So here's just an example from Vision. So this is an example where you increase the time, actually for a certain time, you increase the amount of light until you can just barely see it in the fovea. And what you see is when you plot these seeing functions, this is a log luminance times time, this limb of the function. And it's a constant, see? So this is log L times T. So this is perfectly constant, right up to a knee here. And then after that knee, its luminance is a constant. So the photoreceptor integrates over this period. You can see this. So here's the idea of why it impressed me so much about these functional relationships. If only, and like this LT constant function is known as Bloch's law or Bunsen-Roscoe law. So if only one point had been studied, or maybe 2 points had been studied, it's unlikely this functional relationship, or these functional relationships, would have ever been shown. And to show you how omnipresent these functions are, it's true in the periphery. It's true when you have an adaptation, so this is delta luminance times T. You can see these functions with these E's, you can see this very gradual change in the knee of these functions. So, and it's basic to how photoreceptors work. So, you know, it's one of those kind of things that I really imprinted on it. So, what I wanted to do was to study functional relationships, as Jeff mentioned, color or hue discrimination. And so I designed, this was the area where you didn't have electronics, so you built all these things. So I actually designed all these optics. I studied optical engineering, machined all the parts, so I could get a precise control of wavelength and present it to study pigeons. There was a big controversy there as to whether generalization, discrimination were inverse processes, and this is very related to it. And so in this procedure that I'm going to just briefly mention here, it's the same different procedure with, this is a split field, and different wavelengths can be put on these two halves of this field. So when they were the same wavelength, then pecs to this left P over here were rewarded with a probability, certain probability greater than 0. And I'm going to manipulate that. That's how I'm going to get the receiver operating on RLC functions. And of course, when they're different in wavelength, then this is the different response, and you can manipulate that too. So here are some more parameters of the experiment that you can read about in 600 trials per session, and so on and so forth. And so what I did is to manipulate the probabilities of reinforcement, and for a moderate discrimination, you map out these ROCs and easier discrimination there. And if you plot these ROC functions on normal deviates, z-scores, you get straight lines. So here's an example of results from one of the 4 subjects at one of the reference wavelengths. And so you can see that this is the referent wavelength and only a few nanometers less. You can see this is a difficult discrimination. So you can see these RLC functions. So what do you do with this? What I did with it was to measure discriminability at a neutral point. That's where there's really no bias. The chances of making error the same or different are equal according to the signal detection theory model, where the criterion is right here. And so I did this for all of the birds, and then we're going to plot this and use this to look at hue discrimination. I'll spare you the other 79 figures that look like this. and so when you plot this d prime as a function of wavelength, you can think of this as a second order functional relationship, and I got functions like this. There's that same bird. Here's the first 10 of them. You can see nice linear functions, more or less look into the origin. Well, how does this relate to the pigeon's discrimination? Well, you can see they're discriminating really well here, and much less well here. So this is the whole point. So how do you translate this into these different functions? Well, you can take a criterion performance, like d prime of 2, and you look at the wave number difference. It's like wavelength, just numbers. And you take another point, and you can see it's like 12 rather than 35, much better discrimination here. So you do this at not just one criterion performance, but you do an infinite number. What I did is at 9 different criteria, 20 wavelengths and 4 birds. Here's all the functions from that same guy. And what you do when you average across the birds, you get functions that look like this for different d prime performances. And these dips specify points of good color discrimination. Pigeon can discriminate. Color differences here much better than you or I can. I couldn't check out my apparatus without going and looking at the dials to make sure there was a wavelength difference, wavelength sophisticated, because I could see no color difference at all. You know, I mean, they're just super neat. So, what do these points of good hue discrimination mean? Well, if you turn to human color vision and you look at color naming, identifying the transition between hues, they line up with the points of hue discrimination function, the dips, very well. And here's the human color spectrum to show how they line up. You get a transition between blue and green here, between green and yellow there. So what you get, as you go wavelength by wavelength, nanometer by nanometer, some points have big hue discriminations, and that's where you get the very sharp color discrimination at those points. And I did the same thing with pigeons. I developed a color naming procedure. This was done in a so-called Taj that actually Bill Cumming named it Taj. And a procedure, a categorization procedure to look at points of hue discrimination in this color naming procedure. See how well it lines up with these 2 points of the hue discrimination. So together, these functional relationships show qualitative similarity between color naming and hue discrimination for both pigeons and humans. But there's a quantitative difference. The spectral points of hue transition are different for pigeons than they are for humans. And this is related directly to the photopigments and photoreceptors in these 2 different animals. So what about functional relationships for cognitive process, which is the main point of the presentation here. And I want to talk about these functional relationships for 2 different learning processes, matching sample, pigeons, same different learning and pigeons and capuchins and rhesus monkeys. So in matching the sample, the task is to get sample stimulus. There are 4 groups of pigeons. And the distinction between the groups is the number of responses that they were required to make to the sample before getting the choices. Okay? And so they make the responses to the sample, they get the choices, and they're presented together. And so the different groups of pigeons made different numbers of responses. And since it's matching the sample, this is the correct response and uses grain reinforcement. Now in the study I want to tell you about, so here are the other displays that they were trained on with, you can see dugout and grapes, and so the reason, so this is just one other thing about the apparatus. these pigeons look down on top of a computer screen. It's turned upside down. The idea being that, well, pigeons forage for grains in gravel, and it's a little more ecologically relevant if they're pecking down rather than up. And then you can put the reward on top of the pictures, and you get a tighter bond between the stimulus is correct and food was actually eat off of the picture. So that was the idea. And they learn this task. I'll show you the learning just a bit, quite rapidly. And after they learn, then test them with little cartoons of things that they've never seen before. The idea being, what have they learned? Have they learned an abstract concept? Can they do it with a stimuli? And here are the results. And so here are the different numbers of groups of different groups of pigeons with their different response requirements. Now here are the baseline performances. They learn about the same rates in these situations and perform about the same level. Now the critical difference that I'm going to focus on is the transfer of novel stimuli shown by these darker hatched areas. So just consider the question, if this experiment had only been conducted requiring either no or just one response to the sample stimuli, the conclusion would have been that there's no concept learning. Pigeons don't learn concepts. It's just item-specific learning. In fact, in this case, it was configural pattern learning. But by expanding and putting in this group where requiring 10 responses to the sample, now, if that had been the only point that was investigated, then a conclusion would have been partial concept learning. But by also including this group requiring 20 responses to the sample, as you can see, there's no difference between transfer to the novel stimuli and the baseline performance. So that conclusion, if this were the only test conducted, would have been full concept learning. The pigeons learn relationally and show full concept learning. But together, the functional relationships show, or this functional relationship, by varying the sample stimulus response requirements, shows a continuum of sample response effects that determines the type of learning. Item-specific learning in one case, relational in the other, and relational in this case where you get full concept learning, which is a requirement of concept learning, is to do relational processing. Okay, so same different learning. 3 species, testing apparatus. These are reward areas. The similarities, both had the same video touchscreen. Monkeys had a template. The task was respond to the sample. Pigeons, of course, learning from the previous experiment I showed you, haven't pecked the sample 20 times. Because you want to look at this, try to get, how's concept learning gonna be, again, the same different tasks. Monkeys, they touch it 10 times. Takes oftentimes about the same amount of time. So half the trials are same, and same responses to the lower picture, different responses to this little white square. Other training pictures were these 8. Jocelyn so nicely said all this before. And now you can see the acquisitions, they all acquire about the same rate. Interestingly enough, pigeons even have a leg up early, or a wing up early in training. Even over some of the monkeys, but roughly about the same rate. So after they have learned this task, I showed you the training stimuli. We test them with transfer stimuli embedded in a full 100 trial session. And here are the actual same and different trials for the first transfer test, 5 each, and we tested for 6 days. A little procedure, but there's no concept learning. None of the species show any concept learning. So what do you do? Well, we expanded the training set, took 32 items and tested it. Okay, now we're getting some the concept learning, and then transfer tests with novel items in there. Now we're getting some transfer here. So if the training and transfer tests on these three species have been limited right to this region here, the conclusion would have been that, well, pigeons really don't show any transfer, very little above chance transfer, little or no concept learning, relative to their baseline performance, but monkeys show partial concept learning and partial transfer. So what we did was expand the training set to 6,428 items, and now things are looking even better for all the species. So if the test had been conducted at just this point, then the conclusion would have been, hey, pigeons can partially learn an abstract concept that's different, whereas monkeys fully learn the kind of same different abstract concept. So Undaunted expanded the training set further for pigeons, and now you can see that this performance is even much better. So, of course, if we just tested at this one point right from the get-go, hey, look, pigeons can learn an abstract concept, just like monkeys can. They transfer as well as they do to their baseline performance, as do monkeys. So, together, these functional relationships show pigeons as well as monkeys can learn an abstract concept, which is a qualitative similarity. And pigeons appear to require more exemplars of the rule. In other words, more training stimuli, giving more training exemplars to learn this abstract concept. And this appears to be a quantitative difference. And I say appears to be because there's a special training condition where pigeons can even show quantitative similarity as well as qualitative similarity to monkeys. And this is a condition showed where we took other groups of pigeons. We trained them initially at either in one group with a set of 32 pictures, and the other group a set of 64 pictures. And that's instead of starting with 8, expanding to 16, 32, and 64. So with these experimentally naive guys, we started with just 32. Initially 32 or 64. And when we do that, we get these results here. Now notice, these are the previous results I showed for the pigeons. Notice at this point, moves right up there, right in between the results between the capuchins and the rhesus monkeys. And this point here moves right up there, midway between that for the rhesus and the capuchins. So, in this case, it shows that from these functional relationships, similar pigeon-monkey concept learning, same sets of these are precisely the same sets of training, stimuli, and transfer stimuli. Now, pigeons have a carryover effect, meaning that the reason they didn't transfer up here initially, it's a carryover effect where prior learning restricts the domain for further learning. They're reluctant to expand their domain, and we've referred to this as restricted domain relational learning. Please don't ask me, why didn't you test naive groups of rhesus monkeys and capuchin monkeys and things? Because buying naive monkeys and trading, you can't do this. But it looks like monkeys are much more docile in this expansion of sets is what I would guess. Okay, so now I want to turn to functional relationships for investigative memory processes. I'm going to talk about visual-less memory, auditory-less memory, proactive interference in visual memory, and short-term memory from multiple item displays. Now, for the visual-less memory, I can go through this rather quickly, but let me first show you one of the first experiments that Steve Sands and I did with monkeys. Now, these were in this case, a monkey, Oscar. So Oscar's two, but this is Oscar. Sat in a chair, he had a lever, he pushed down to start a trial. And after he did so, then he would get a list of items, up to 10 items. I'm going to show you 10 on a list. He actually can perform accurately with lists of 20 items, which is kind of, at least initially, taps kind of the limit, you know, and how accurately we can perform with these kind of travel slides that we use. So he gets a list of 10 items, and if the test item, we get a single test item after the list. If it matches any one of the list items, then in this case it moves the lever to the right, and if he does so, he gets rewarded. And half the time it won't match any of them, you got to move the lever to the left to get your reward. So, and what you get is, here's a comparison of the monkey and human with these same items and same training criteria and everything. So this is the first primacy effect shown for any non-human animal. And that's important, as Jocelyn mentioned, because it was thought to represent the long-term memory. From Atkinson and Schifrin, you get the dissipation and recency effect with these long lists in humans, been shown for a long time, thought to represent short-term memory. Items come into short-term memory. If it's sufficiently rehearsed, it goes to long-term memory. And the first list items have more opportunity to be rehearsed. And that's why you get this kind of long-term memory deal, and there's some other evidence as well. So, to further investigate the serial position function effects, the primacy and recency effects, these serial position functions, of course, are functional relationships of primacy and recency effects, and to investigate them, use these short 4-item lists that Jocelyn told you about. And one of the reasons is that if you use longer lists, some of the changes in primacy and recency effects that she showed take place before you've even finished the end of the list. So by using a very short list, one can look at the transitions in these serial position effects. So since she just went over it, I'll just bypass this. Thank you, Jocelyn. You saved me a lot of time. I was worried whether I was going to get through this. You know, she says, it's unfair on French. I died doing it. Okay, so systematically varied retention delay for capuchin monkeys. So these are capuchin monkeys. She showed you rhesus monkeys. So you can see this very strong recency effect. Primacy effect comes in later, just like she showed you on the previous result. And so changes amongst the serial position functions, which are functional relationships, they change with retention delay. Think of this as a second order functional relationship. Rhesus monkeys, these were some of the first we did, both with the monkey sitting in the primate chair and moving a lever with carousel projectors that Cook mentioned, as well as the video touchscreens. And pigeons, Jocelyn showed you these results. And humans, the way we've tested humans with these short lists, so we would get ceiling effects, where to use kaleidoscope items, as you can see, some of these here, you know, which make a really nice way to test. And you look at these in more detail, you see these strong recency effects, primacy effects come in, and recency effects fade. So you get these transitions over time. But notice that monkeys make these transitions in 30 seconds, pigeons in 10 seconds, and it takes about 100 seconds for humans for these functions to make these transitions from a very strong recency effect to a primacy effect. And Endel Tolving, in his chapter in the Festschrift book in honor of Gordon Bauer, his chapter was termed The Law of Primacy. And he talked about some of these results as well as others. There's law of primacy after this delay, this primacy effect. So together, these functional relationships show qualitative similarity. You've seen these statements before. I'll just say them again here. Qualitative similarity across species in visual memory, but the different rates of change of these serial position functions, which are really indicative of some underlying processes how memory evolve in these species. This is a quantitative difference in visual memory. So we can ask the question, is memory similar in other modalities? And I'm going to show you some brief examples from auditory memory. So the auditory memory tasks that work for us, remember Bob, when we were trying to translate this, we had several false starts. And here's the one that worked. So they're going to get a list. I'm just going to tell you the final deal on the test. List of fourth sounds, large set. Here are some parameters. Here are some examples of some of the sounds. So they get a list of fourth sounds, and then after a delay, so we have a delay because that's going to be the parameter, then they get the same sound presented from both side speakers at the same time. It's not two different sounds, it's the same sound. And if that test sound matches one of the list sounds, the guy needs to go over to the right and touch the right speaker. It's actually a little copper screen in front because we put a little high impedance CMOS circuit on it and tell it to touch it. So go over and touch that. And if it matches none of the list sounds, which you will have the time, it has to go over to the left. So here are the kind of results we get. So you can see nice serial position functions, primacy and recency effects. You look at this 10-item one, remembering what Oscar had produced in visual memory with this 10-item list. It doesn't look too dissimilar. Maybe a little stronger primacy thing going on here in these longer lists, and in some cases maybe not relatively quite as strong a recency effect. And that'll become even more apparent when I show you what the serial position functions look like for auditory memory. So for auditory memory, here's what we got. Now you can see this starts out a very strong primacy memory effect, whereas there's no primacy effect for the visual memory. And this strong primacy memory fades to the chance accuracy as we go out to 30 seconds. Whereas the recency effect is weak recency effect, and that strengthens when we go out to high accuracy. So just the kind of opposite changes that you see in visual memory, and to test some of these processes that produce these serial position function changes, as Jocelyn mentioned, we're going to look at retrieval inhibition, or inhibition among some of the items. And one of the strongest, we had many experiments on this, one of the strongest demonstrations of this is, so these comparisons were only made evident by testing these functional relationships across a whole range of delays. That's the whole point of the functional relationship here. So in order to test some of the processes that are going on here, what we did was we tested single item memory and compared it to the last or fourth item memory performance. This is important because you can think of that comparison is just like the 4-item list memory task, but with the first 3 items removed. So when we did this, here are the results. You can see there are good performance here for single item list memory, and relatively poor performance for the fourth item list memory. And when we look at that difference, that's the difference you see here. So this difference, or gap, shows proactive inhibition from the 3 prior list items on retrieval memory of the fourth item. So retrieval of memory of the fourth item was inhibited by having these three items preceding. And you can turn this around. The comparison isn't quite as precise, but precise enough to looking at this gap here, and you compare this single item memory to the first list item, and you can see the retroactive inhibition from the last three items on retrieval memory of the first item. So together, these functional relationships show qualitative differences between auditory and visual memory, rhesus monkeys, inhibitory processes among auditory items that affect retrieval memory. And none of these findings or conclusions would have been made without testing memory over a substantial portion of the range of delays to generate these functional relationships. So let me tell you about functional relationships in proactive interference of memory. Proactive interference is the interference of items on previous trials with the current trial performance. And in these list memory or same different tasks, the locus of PI occurs when the test item, that's the test for proactive interference, it's always on a different trial, but that test item differs from the sample item on the current trial, the test trial. But that same test item had been seen as a sample on a previous trial. I'll show you a diagram of this in just a moment. And PI, proactive interference, is endemic to all tasks with repeated stimuli. In order to investigate proactive interference and how it affects memory, one needs to use baseline trials that are trial unique, meaning that you don't repeat the stimuli from trial to trial. Because if you have a small set of stimuli that were repeated a lot, what happens is proactive interference accumulates over time. Kepler and Underwood showed that in 1960s. Accumulates over time, and it saturates, and proactive interference becomes really high. So if you want to systematically test how items back in time can influence later performance, all of the other trials except these proactive interference test trials need to be trial unique. So you can test the effects against this baseline of no interference. Now, when this is the same monkey that moved the lever in the visual list memory task. So what we did was we tested him large sets of stimuli on all the baseline trials and put, in this case, in the previous trial, one of the 10 items was going to appear on the test trial as a test item where that test item didn't match any items in the current trial. So the correct response was to say different, but having seen it in the previous trial confuses the subject. In fact, it confuses and drops performance like over 35%. But you can see as it recedes back in time, so this is the second trial, items 21 through 30, you can see there's less proactive interference and so on. But still, items that were seen, as many as 60 items previously, still produce a 10% proactive interference effect relative to baseline no interference. So that's far back in time. That's, you know, that shows that things are going way back in time. So another way to test it, rather than with list memory, so simpler, is to test these same proactive interference effects and generate similar proactive interference functions. And we did this with pigeons, and this is a single item memory experiment rather than a list memory experiment. Here's some just parameters of the experiment here. So this shows, this is the interfering sample stimulus. And this is the trial n-1, that's the trial prior to the interference test. So here's the interference test stimulus. So you can see it doesn't match the bad item, and the guy should, the correct response is to touch this little white rectangle. But having seen it as a sample in just the previous trial confuses the subject, and the chances increases the chances they will say, it matches the sample that I'm supposed to remember. Okay, so what we did was test pigeons. We tested them under 2 delays. So even with a 1 second delay, you can see there's a substantial proactive interference effect at 1 and 2 items prior. It dissipates relative to baseline. But notice when we do a test with a 10-second delay. With a 10-second delay, here we get this whopping 47% proactive interference, which, as you recede back in time, gradually dissipates, but still very large proactive interference relative to the baseline condition, even at 16 trials prior. Now, one of the things that, if you think about it, is counterintuitive here. Now, these are tests, all of the trials have this. This is block tests, as I mentioned. All of the trials have this 10-second delay. So all of these items are being seen much further back in time than these items are being seen under the 1 second condition. Further back in time should mean more forgetting. Further back in time, something occurs, you have more decay, more forgetting, this decay notion of memory. But if memory is decay, you can't remember the interfering stimulus. If you can't remember the interfering stimulus, you're not going to have any interference. See, this is a way to tell how well a subject remembered something back in time through its interference on its current trial. So you can go way back in time and say, how far back in time can you remember? But look at this counterintuitive effect. So, like in this condition, items were seen as much as several minutes earlier than further back in time than these items were. But yet there's this much greater interference effect. So, and that's just what I said here. So, what's the resolution? Well, the resolution is that proactive interference depends upon time ratios, not any absolute time, how far back in time. Okay, so time ratios. Well, you've seen signal detection theory models before, you're going to see another one. So here's internal representation of elapsed time. This is the time of the interfering stimulus, the time of the current sample, the current stimulus, the noise. So this is what you're trying to discriminate. When was that seen? Was it seen further back in time? And so on a log scale of time here, we model the results. The critical parameter here is this ratio of times. Time to the current sample divided by time to the interfering sample. And you can think of this, there's this telephone pole of metaphor of memory. So as you increase the delay, that first telephone pole is going to be further back in time than it was with a 1 second delay. And then the other telephone poles, these are the items that you're trying to remember back in time, appear relatively closer. This is the log scale of time. It's fit by 2 parameters, but both functions were fit by the same 2 parameters of bias and asymptotic performance. The fits are very good. The functional relationships show good fits, accounts for 95% of the variance. Counterintuitive finding of greater proactive interference for longer delays depends upon a time ratio. This is Faber-Fechner log time scale. Some of the implications, I'll mention 2 of them. One is that proactive interference doesn't depend upon absolute time. So this rules out familiarity. Familiarity is an absolute time back in time. So proactive interference depends upon a ratio, not an absolute time. Another implication is that a lot of us use timeout. In fact, several times during our conference already. If you make an error, you get a timeout. Timeouts for errors. And the idea was that you would decrease, the opportunity for reinforcement because you would have longer to wait. So that was one of the current explanations. But timeouts for errors would reduce time because it would change that ratio, decrease that ratio, in addition to any punishing effects for delaying reward. I'm going to end here by talking about functional relationships for short-term memory processes using multiple item displays. So here's shows you the procedure. Subjects, monkeys, and humans in these studies. This is a familiar game. You get maybe a display. This is Caitlin's dissertation work. Terrific. Get a display of 5 items, for example. Then after a delay, what item changes, and you got to identify the item that changed by touching it. And then you have an intertrial interval, and then it's variable number of items. Let's say on the next trial you get 6 items, then you got to identify the item that changed. So large item sets, in this case clip art, 976 clip art items. Here's a picture of Cisco working on the task. And here are the results. So present correct. You can see as the display size increases, performance goes down. Here are the monkeys. They perform less well than humans do, not surprisingly, but still, this is under these conditions, it's still, above or right at 80%. Now, this is somewhat better performance clip art rather than the colored circles or squares that we also tested them with the same colors. Humans have slightly better performance there with clip art. So here's the deal. You get these nice relationships. You get these nice relationships. So how do you compare and relate these species on their short-term? Well, you're going to see another signal detection theory model, I got to tell you. So here we have, we transform our percent correct scores to D prime in this way. And then according to signal detection theory, we fit these functions, and these functional relationships were fit by an inverse power law. And this is specified by the falls out of the signal detection theory model, where we get good fits to these functions, counts to this to R squared values. And together these functional relationships, the model suggests memory is noisy versus all or none fixed capacity. Noisy memory is plausible, much more plausible than fixed capacity from a neural perspective. It's distributed as a continuous resource among items. It doesn't have to be every item gets the same resource. Fixed capacity is you either know it completely or not at all. And according to this framework, monkeys and humans show qualitatively similar short-term memory processing. There's also a quantitative difference that humans perform more accurately than monkeys do. So there's this quantitative difference. So some concluding remarks here. concluding remarks. So in this presentation, I've argued for the importance of functional relationships and comparing cognitive processes. You've seen these from some of our presenters. A nice symposium, a terrific presentation today. And I hope I've succeeded in some measure in conveying the importance of testing functional relationships to compare cognitive processes. And these tests of functional relationships were presented across a range of values of one or more critical variables from psychophysics, from learning, and from memory. And the functional relationships were compared within and across species, showing qualitatively similarity of cognitive processing and some quantitative differences. And I'll emphasize again that none of this would have been possible if tests had been conducted at a single value or even two values. Moreover, conclusions, if they'd been conducted at just a single value, would likely have been misleading. So also, none of this would have been possible without the terrific support and participation of collaborators that I've been so fortunate to have over the years, and none of it would be possible without the support I was so fortunate to obtain from NSF and NIH and other agencies. And possibly none of it would have been possible without the guidance from my mentors and sponsors early in my career. I've stayed in touch over the years with Gordon Bauer. He's apparently liked the work I've done and complimented me on it. He even invited me to this festschrift that was held a few years ago, along with his 55 PhD students that he mentored over the years. I was just an undergrad. So we had a celebration for he received the National Medal of Science, and we had a celebration for this a couple of years ago. And there's him, of course, receiving the National Medal of Science. And at that celebration of his, the book on his festschrift had just been published, just prior to that. And this is his book, Mine. And so during that celebration, he signed my flyleaf to me. He said, "The first student is always the most memorable." Now, I mean, this is really the law of primacy, I gotta tell you. And it's in the same book. I think chapter 2 is by Ethel Tolving, The Law of Primacy, so it's the same book also. Thank you.
Questions, and then we'll go after if there are any for Dr. Wright.
Yeah, can everyone say, I know, Ed can talk about things. Yeah, of course.
Thanks, Tony. We'll see you all in a bit.